Hi student, good morning. Today we are going to discuss topic number 17. The title is inheritance. In this subtopic, we have to discuss the difference between the inheritance and the variation. So what is inheritance? Inheritance means the parent characteristics are transferred into the into the offsprings, due to which offspring look like their parents. But some of the characteristics, either not transferred or are hidden in the offspring, due to which offspring look different from their parents. So this is called as the variation. So now come to the first slide. We just want to review the some small definitions. For example, inheritance, you can see the transmission of the genetic information from parents to the offspring. Now, what is chromosome? Chromosome, is a thread-like structure of DNA that carries genetic information in the form of gene. Because in human being, there are 46 chromosome, and each chromosome has some specific gene, and the genes are responsible to make different characteristics. Now, what is the specified definition of gene? A gene, is the length of DNA that codes for a particular protein. Now, what about the allele? One of the number of different versions of a gene. Means each, like the gene has some versions. Okay, so the version of the gene is named as allele. Genotype, the genetic makeup of an organism consisting of all the alleles present. Now, what about the phenotype? That is the observable features of an organism as a result of expression of particular allele of the gene. Now, homozygous, an organism containing two identical alleles of a particular gene. Heterozygous, an organism containing two different alleles of a particular gene. Diploid nucleus, nucleus which contain a full set of chromosome, that is 23 pairs. 23 pairs mean 46 chromosome. And if the nucleus has a half number of chromosome or one set of chromosome, means 23, so it is named as the haploid nucleus. So you know that in human being, there are like 23 pair of chromosomes in the normal body cells. But if you're going to study the reproductive cells, so these reproductive cells contains 23 chromosome. Means the person get 23 chromosome from father in the form of sperm, and 23 chromosome from mother in the form of egg cells. And in male, there is a heterozygotes, mean the uh, chromosome XY, but in the female there is a homozygotes, they are XX. So the question arises that who is responsible to determine the sex? Of course, like the male or the father is responsible to determine the sex, as the father has XY chromosome. Here you can see that the chromosome is there, and chromosome contains so many genes, and gene is the length of DNA. So you can also see the DNA, that is the double stranded molecule. Now protein synthesis, see the gene, gene responsible to make the like specific characteristics. But how these characteristics are made? So firstly, you should know that what is transcription. Transcription means the formation of RNA from DNA, and from the RNA, then protein is synthesized. Specifically, three types of RNA play very important role. That is messenger RNA, rRNA, and tRNA. So DNA controls the function of the cell by regulating the protein synthesis, antibodies, and the receptors for neurotransmitters. Protein synthesis is important to maintain cell structures as well as the producing the enzyme in catalyzed metabolic reactions. The gene which codes for the protein is used to make an mRNA copy in the nucleus. mRNA is similar to the single strand of DNA, although contains the base U rather than T. U mean uracil, and T mean thymine. because in RNA, instead of thymine, there is a uracil. The messenger RNA copy leaves the nucleus and travel through the cytoplasm to ribosomes. The ribosome uses the messenger RNA strands to produce a chain of amino acids which make up the protein. The order of the amino acids chain is determined by the order of bases on the messenger RNA. This order is specified to a specific, protein. As each type of cell has a different functions, not every type of protein is synthesized in every cell. The genes to code for each protein are present in every cell. However, these genes are therefore switched on or off. When the gene is switched on, it is expressed. It is expressed means it's going to make a specific type of protein, and this protein is going to make a different type of characteristics. And the protein associated with the gene is synthesized. When the protein is not required, the gene is switched off. Yes, if like some specific type of protein is not required, then the gene is switched off. Switch off means it's not going to perform its job. Okay, now here you can see there is a structure of the DNA. As you know very well that DNA consists of three particular things. What are these things? The nitrogen bases are there, and deoxyribose sugar and phosphate group is there. In the mid, you can see that thymine and adenine, they make a double hydrogen bond, and cytosine and guanine, they make a triple hydrogen bond. So here you can see that in each strand, one nitrogen base is there, which is joined by the other. Okay, and with each nitrogen base, there is a attachment of uh, sugar. Which type of sugar is this? This is a deoxyribose sugar, and then the phosphate group is attached, and one nitrogen base, one phosphate group, and one sugar is named as the nucleotide. And when so many nucleotides combine together, they make a polynucleotide. So on each side, there is a polynucleotide chain. And when these two chains, are just coiled together, so it makes the DNA, because, DNA is a double-stranded molecule. But RNA, RNA is single-stranded molecule. So the process in which the DNA make the RNA called as transcription. And this process takes place inside the nucleus. And then the messenger RNA, enters into the cytoplasm. Then there is a long efforts, okay, of different type of RNA like messenger RNA, tRNA, and rRNA. Then there is an attachment of amino acids, okay, on the messenger RNA, and it makes a long chain of amino acids, which is known as protein. Now, we also discuss in this chapter, these are division of cell, for example, like the mitosis. What is mitosis? Mitosis is a form of cell division. During mitosis, nuclear division of a parent cell occurs, producing two genetically identical daughter cells. Right? Identical means both the cells have the same number of chromosomes. For example, human cell has 46 chromosome. When it's going to divide, so it's going to make two cells having the same 46 number of chromosome. Mitosis is used to create a new cells in the body of a body to repair and replace old and damaged tissues, as well as allowing growth of the organism and playing a role in asexual reproduction. Mitosis involves the splitting of chromosomes into their two halves, each of which are known as chromatids. Okay, each of which known as the chromatid means the one chromosome is made by the two threads, and each thread is known as chromatids, and these two chromatids joined by a point that is called as a centromere. Before replication, application can occurs, the chromatids in the parent cell must replicate, mean make its copy, to produce the identical copies of themselves which pair as chromosome. The chromosome line up along the nucleus. The chromosome separates so that Each identical chromatid is pulled to opposite poles of the cell. The cell membrane constricts in the middle and the nucleus is separated as the cell splits into two new daughter cells. Each of these cells will contain the set of chromatids which then replicate themselves again to produce a new chromosome. So this is how, like, the new chromosome are made. Okay, now see here, mitosis. You can see a pair of chromosome. You see the maternal chromosome and the paternal chromosome. And here, it's going to be replicated. You can see here inside the cell, the nucleus is there. Inside the nucleus there is a chromosome, and here you can see that how, like, the replication take place. See here, the each chromosome has been duplicated. The nuclear membrane breaks down and the chromosome line up along with the center of the cell. Here, in IGCSE biology, there is no detail of the different phases of mitosis, because there are four phases like prophase, metaphase, anaphase, and telophase. These phases detail are not given here. So we are just going to move to the another topic, stem cells. Now, what are the stem cells? Stem cells are found in embryos or in the bone marrow. These cells are unspecialized and divide by mitosis to produce daughter cells, which then specialize to have a variety of functions. Cells once specialized cannot produce unspecialized cell. For example, a cell which makes up the hard tissue cannot divide to make a cell which can act as a skin tissue, as the cell produced will already be specialized in the hard cell. See here, like, stem cells, has the capability to make any type of cells. Okay, so this is stem cell, they are the unspecialized cells which has the capability to make any specialized cells. For example, like, specialized cells mean like the sponge is made. Okay, specialized cell mean the white blood cells are made. Specialized cells mean the cellular cells are made. So but stem cells are unspecialized, and that the cells has, the nucleus has chromosome, and chromosome, has the gene. So some genes, are switch on or off, okay, due to which, like, the type of the cell is to be decided. Okay, now meiosis is used to make four genetically unique daughter cells and is used in the production of gametes. See, if you are asked, like, what is the difference between the mitosis and meiosis? Mitosis is the process in which one cell divides into two cells having the same number of chromosomes, but meiosis is the division of cell in which one cell divides into four cells having the half number of chromosomes. And in mitosis, there is a process of growth and repair, and meiosis, this is a process of formation of gametes. As you can see here in the diagram, whatever we have discussed it, okay, it can even seen in the diagram. Now come to the see the difference. Two cells produced known as the daughter cells and four cells are produced known as the daughter cells. Daughter cells are diploid, and here meiosis daughter cells are haploid. Daughter cells are genetically identical, but here daughter cells are genetically different. One cell divides division occurs, here two cell division occurs. Okay, now sperm cell determine the sex of offspring. As you can see, like, the mother has XX chromosome, that is homozygotes, and the father has XY chromosome, that is a heterozygotes. So the X is going to combine with the X. Okay, so see XX, and here X combined with X, so XX, and here Y is going to combine with with X, so XY, and here Y combined with X, so XY. So there is a 50% chances of the boy, or the 50% chances of the girl. Now, what is monohybrid cross? Monohybrid cross, is a cross in which there is an inheritance of one characteristics. That how one characteristics is transferred from parents to the offspring. So this is, we call as a monohybrid cross. For example, if you are just stating the height of a plant. Okay, that one, like the tall plant when is going to be crossed with a short plant. So which type of plant are expected in the office springs. So as we are discussing only one characteristics here, so this is being named as the a monohybrid cross. And whenever there is a same type of gene, okay, same type of gene are there, so this is called as a homozygous, mean the a pair of identical gene. If the pair of non-identical gene is there, this is called as heterozygous. For example, capital T, that is homozygous, or small t, that is homozygous. But capital T, small t, it is a heterozygous. Okay, it means non-identical, two non-identical gene. If two identical homologous individuals are bred together, it is referred to as pure breeding, and the offspring will have the same characteristics as the parent. Breeding heterozygous. Okay, parent is not pure breeding, as there are a number of different alleles that offspring could display in their phenotype. Now, what about the dominant recessive allele? Dominant allele, even if it is a single, it's going to make the phenotype. But recessive allele is that allele which can make all the characteristics which can express itself when it is in the form of pair. Now come to the next one. See here, yes, you can see if there is a cross between the tall plant and the short plant. Tall means this is a pure tall. This is homozygous dominant. So in F1 generation, we will see that all the plant are tall. See here, capital T, small t, it means like, this capital T, is a dominant, small t is a recessive. So the person is going to be tall, means 100% kids would be, tall here in F1 generation. So see, if you are talking about like, the plants, see all tall plant are there. Now, if F1 is going to be, F1 generation is going to be crossed with F1, and because in F1 we get heterozygotes. Okay, so heterozygous is going to be crossed with heterozygous. So firstly, like, the gametes are made, okay, and then, capital T, small t are going to be segregated. After that, they are going to be combined independently. For example, see, capital T is going to combine with a small t, okay. Capital T is going to combine with capital T. So see, capital T, capital T small with capital small t. So capital small t. Here the same, small t combined with capital. So capital small t, and small t combined with small t. So they make. So actually, here, what is the ratio that we get it? See here, if you're asked like, how many tall are there? So see, three tall and one dwarf is there. In three tall, one pure tall and two impure tall. Mean one homozygous dominant tall is there, and two are heterozygous tall. But one, one is dark. Means if you are asked the percentage, so see, 75% are tall and 25% chances of the darkness. See, 1 out of 4. So the ratio that we get it here, is 3:1, because 3 are tall and 1 is dark. 75% tall and 25% dark. So ratio is 3:1. Now, there is also one more cross. This is we call as the test cross. If you just want to know that either this plant, as it looks tall, it is really tall or not. So when it's going to be crossed with a homozygous recessive, then we are going to get like 50%, 50% tall and 50% tall. If we get 100% tall, it means this person is pure tall. If we get 50% tall and 50% dark, means the ratio is going to be 1:1. So it is called as a test cross. So see, a plant breed has a tall plant of the unknown genotype. How can they find out whether it is a homozygous dominant or the heterozygous? So this is, this can easily be find out by the test cross. Okay. Now, there is another type of dominance. This is called the codominance. Inheritance of blood group is an example of codominance. There are three alleles of the genes governing this instead of the usual two. For example, allele IA and IB are codominant. Mean the blood group, is AB. The blood group AB is the example of codominant, but both are dominant to IO. I represent the gene and the subscripts A, B, and O represent the allele. For example, IA result the production of the antigen A in the blood, and IB result in the production of the antigen B in the blood, and IO result in no antigens being produced in the blood. These three possible alleles can give us the following genotypes and phenotypes. Now you can see here the genotype. Actually, you know what you have to learn? You have to learn that the different blood groups, have which type of genotype. For example, blood group A, has two genotype, IA and IA and IA and IO. And blood group B also has two genotype, but blood group AB has just one genotype, and blood group A also has just one genotype. So if you are asked, like, that how, like, uh, the offspring are going to have the blood group if the parents have this type of blood group, so you have to cross with their genotypes. For example, see here, if the mother is heterozygous blood group A and father is heterozygous blood group B. So what are the chances of blood group in the offspring? So you can see here that IB IB is going to combine with IA, so it's going to make IAIB. So mean the blood group is going to be AB. If B is going to combine with IO, so it's going to make IB, IO. So it is the blood group of B blood group. And if IO is going to combine with IA, so it's going to make IA, IO, that is blood group A. And if IO is going to combine with IO, so it's going to make IO, IO means blood group O. So if father and mother both are having different blood group, like father has B and mother has B, and both are in heterozygous condition, so we get 25% chances of each type of blood group, as you can see here. Now, sex-linked characteristics. Some genes are located on the sex chromosome, characteristics which come from the parents. These genes is referred to as the sex-linked inheritance. See, the genes are found on the X chromosome, and this X chromosome is common in uh, male and female both. Okay, so male, has XY chromosome, and female has XX chromosome. So as you can see here that XR, okay, and Y, that is the father, like, father chromosome have these genes. So when father gene is going to combine with the mother gene, so you can see here XR, when combined with XR, so XR female normal, and XR XF, so female carrier. Because female has, like, same type of chromosome, XX chromosome. So when, like, uh, there is a presence of a particular gene, only one X chromosome, that person is going to be the carrier. Okay, but as, like, the male has only one X chromosome, so if male has the defective gene, so is going to have that particular disease, either it is a color blindness, hemophilia, okay, or any other disease. So this is about the genetics. Okay, we have discussed the inheritance. If you have any confusion, you can ask me. Okay, my WhatsApp number is there, my email address is there. This video is also available on the YouTube and also available on the TikTok. So if you want to ask anything, you can give the comments, okay, and you can ask any question that you want. Okay, okay, thank you very much. Bye.